Okay, so now we're in our third and final stage of how we make proteins from DNA. And that name of the stage is called translation. And remember I said it begins with our DNA being housed in the nucleus, and we have to make a copy of that DNA into something that we call mRNA or messenger RNA. That messenger RNA can travel into the cytoplasm where it now is going to be translated. So translation occurs in the cytoplasm, and it occurs in 3 stages as well. So it's in the cytoplasm, and there's 3 stages, and we're just going to look at each stage in a little bit more detail. Very, very similar to transcription. Remember, transcription had an initiation, elongation, and termination process. The same 3 stages are used to represent what's happening in translation. So if we can picture, we have ribosomes. Remember, in our cell parts, ribosome was one of our cell parts. Now we're going to look at how it helps make proteins. So that was the job of the ribosome, just generally, but now we're going to see how it actually does that. So the mRNA transcript has just traveled into the cytoplasm, and the ribosome will come along and bind to that mRNA transcript, okay? And I'll just show you what this looks like. So this big green thing would be representing our ribosome, which is made up of a smaller subunit and a larger subunit. And basically what it's doing is it's providing a large site for where proteins can actually be made. So just let's go back to Unit 1. Remember that proteins are made up of a bunch of amino acids that bond together. So what we're doing right now in the process of translation is bringing those amino acids one by one as we read our mRNA transcript. So if I can put together this picture, this big long thing is our mRNA transcript. So if we were to zoom in on that, each of these set of 3 bars would represent our 3 letters code that would code for a specific amino acid. This big ribosome is basically just the factory that allows this protein to be, that moves along this mRNA transcript and allows the amino acids to be joined. Okay, so within our ribosome, we have 2 different sites, and these are important. We have a P site and we have an A site. And I've defined those down here. So the A site accepts something called a tRNA. So remember we talked about the different types of RNA. We have mRNA, tRNA, and rRNA. So tRNA, they're floating in the cytoplasm. Okay, here's an example of a tRNA. So a tRNA basically is an RNA transcript. So this big long thing is made up of different codes in itself. But at the very bottom of it, it has 3 very specific codes that represent that protein. And so at one end of our tRNA, so let's just label this here. This little diagram here is a tRNA. And the components of a tRNA are at the one end, or the top of it, is the amino acid that matches the code at the bottom. And the bottom of this tRNA, we don't call it a code, we call it an anticodon. And that's because it's complementary to what we read on our actual mRNA transcript. So if we had our mRNA down here, this is complementary to what we're reading on our mRNA. So what this would match up with, if this were our anticodon on our mRNA, so let's just give you an example of what that looks like. If we were to have our mRNA transcript down here, each tRNA has its own amino acid that it codes for, and the anticodon is what matches up with what's on the mRNA. So the mRNA for this one would be A, T, and G. This would represent the mRNA and each tRNA. So the job of each tRNA is to come along the mRNA and recognize each set of 3 letters. And there is one tRNA for every single amino acid out there. So this is what the one for methionine, or MET for short, looks like. And remember, if we were to go back and use the genetic code, these 3 letters should code for methionine. But in order for something to recognize it, it has to have the complementary letters for those. And that's why they use the word anticodon. This is our codon, and anticodon would be complementary to it. So this little tRNA would come in, read along the mRNA, and it would match up with these letters, and it would tell the ribosome that methionine is the next one to come in. So the tRNA is basically what delivers all of the amino acids. And there are 20 of them with each their own amino acid floating around the cytoplasm ready to complete this process. Okay, so if we think about the big picture, we have a ribosome. I'm going to make this picture just a little bit smaller so we can fit it on one screen. Okay, so we have our mRNA. This big green thing's a ribosome. And the P site and the A site is where these tRNAs who are floating around the cytoplasm are going to come in and help deliver the amino acids. So you can see at one end of each tRNA we have specific amino acids. And we need to add them on to this growing amino acid chain because remember, proteins are made up of amino acids. DNA tells us the order of the code for each amino acid that will get added in the specific order that the DNA tells us. So these guys come into the specific sites to basically just deliver the amino acid that they're told to deliver. And how they know it's their turn to come in is based on the code that happens along the mRNA. And so in detail, remember, the code along the mRNA will match up with each tRNA's anticodons. And then at the opposite end of these anticodons is the amino acid that will be delivered. Okay, I know that's a lot to take in, and you're going to get some practice with this, and you're going to see it all put together at the end. So there's specific jobs. The A site on our ribosome accepts the incoming tRNA that carries the next amino acid to be delivered. The P site is for the tRNA that is forming the peptide bond with the amino acid from the A site. So basically the P site is there to form that bond for the next incoming amino acid. So this comes in, delivers its amino acid, then it leaves. The next one comes in to the A site, delivers, joins, then leaves. The next one, and the order that this happens in is all determined based on the letters that occur along this mRNA. So the P site is there for the original one to basically join new amino acids to the next growing chain. The A site is for all the new incoming tRNAs to deliver that amino acid. Okay, so that's the difference. You should know the difference between the P and the A site, definitely, and what their job is and how it differs. Okay, the first TNR carrying methionine begins the process by sitting on the P site. All of the other remaining tRNAs arrive at the A. So remember we said that the very first tRNA that's going to come in, or the very first amino acid of every single chain, is always methionine because that is our start codon. That guy is always going to come in and sit on the P site, and it stays there. It's the new tRNAs that are coming in to join their existing amino acids that use the A site to do that. Okay, so recall at the top of each tRNA is an amino acid. There are a minimum of 20 tRNAs, one for each amino acid. Remember, there's a total of 20 amino acids, but there can be up to 64 depending on the species. So if you remember, if you go back to my genetic code note and we looked at the different possibilities of what the three codes can code for, you'll notice that a set of 3 letters, or a codon is the term we use, can actually code for more than one amino acid. We'll look at examples when we get to that. Okay, so that sort of sums up the process, the very first stage of translation, which we call the initiation. So we want to get the ribosome in, we want the start codon, methionine, in the P site, and then each new tRNA coming along is going to add its own amino acid. And that really basically explains the elongation process. The word elongation is used because we're elongating this polypeptide chain. We're just continuing to add amino acids until we have the protein that we want to try and make. So that really summarizes what's happening with elongation. Once the tRNA carrying methionine is on the P site, the ribosome basically just slides along the mRNA and reads each codon at a time. And once the codon is read, the tRNA with the anticodon arrives at the A site and it delivers its amino acid. So this is the continuous process of what's happening. You can see here's methionine. This would be methionine here. It's opposite to our last diagram. So methionine is our start codon. It comes in, it stays in the P site the whole time, and it houses the growing polypeptide chain. And the A site is where the new tRNAs come in just to deliver their amino acids that belong there. And then finally, we're going to reach a stop codon. As that ribosome slides across the mRNA, it's going to reach a stop codon. Remember, there were 3 different stop codons. And the ribosome is basically told to stop at that point. A specialized protein called the release factor binds and causes the ribosome to fall off, and the newly formed polypeptide chain and the mRNA transcript are also released. So now we've got our new protein that we wanted to make for whatever reason our body needed it for, and the mRNA transcript and ribosomal units can be used again to make more proteins. So this whole process can go over again if need be.